Something fires in your brain within minutes of the first bite, long before a single calorie has been digested, absorbed, or turned into blood sugar. It's not what most people assume, and once you know what it actually is, you'll never look at a meal the same way. Ask most people why they stop eating, and you'll get some version of the same two answers. Either the stomach fills up like a container reaching its limit, or blood sugar climbs high enough that hunger simply switches off. Both explanations line up with what people can feel and observe about their own bodies, so it makes sense that almost nobody has ever questioned them. The story about a filling stomach feels obvious because eating a large meal does produce a real physical sensation of pressure, especially near the end, when a second helping suddenly feels unnecessary. The blood sugar story carries its own intuitive logic too. Everyone knows that sluggish, satisfied feeling that shows up an hour or so after a big plate of pasta or rice, when energy finally seems to have landed. Neither explanation is wrong exactly, but neither one comes close to answering the actual question this video is built around. What fires first? Long before your stomach comes anywhere near capacity, and long before glucose has made its way into your bloodstream, something else has already sent a message to your brain, and it starts working within minutes of your very first bite. Start with the blood sugar theory, because it's the easiest one to test against real data. According to a 2001 report from the American Diabetes Association published in Diabetes Care, blood glucose doesn't even begin to rise until roughly 10 minutes after a meal starts, as the digestive system works through the early stages of carbohydrate breakdown. In most healthy adults, glucose doesn't reach its peak until somewhere between 60 and 90 minutes after that first bite. That timeline creates an obvious problem for anyone using blood sugar to explain early satiety. People routinely report the first sense of fullness within 5 or 10 minutes of starting a meal, sometimes before finishing even a modest plate of food. If glucose is still climbing slowly in the background at that point, it can't be the signal responsible for what's being felt in real time. The chemistry is simply too slow. This gap between subjective experience and glucose physiology is exactly why researchers stopped treating blood sugar as a standalone explanation for meal termination decades ago. Something faster has to be doing the early work. And that faster mechanism turns out to have nothing to do with sugar, digestion, or absorption at all. It has to do with a sense most people never think of as being part of eating. The organ responsible for the earliest fullness signal is the stomach, but not through digestion. It works through mechanics. The stomach wall contains specialized nerve structures built to sense stretch and tension, not the chemical content of what's inside it. Researchers Pauli and Phillips, whose work has been foundational in this field, established that the stomach has essentially no chemoreceptors capable of detecting nutrients directly. Its primary sensory job is physical, not chemical. These structures are called gastric mechanoreceptors, and two subtypes in particular, intramuscular arrays and intraganglionic laminar endings, sit directly in the muscle layer of the stomach wall, positioned to detect exactly how much the tissue is stretching as it fills. A 2016 study published in Cell by researcher Williams and colleagues imaged these sensory neurons directly and found that stomach expansion of just 300 milliliters, roughly the volume of a small glass of water, was enough to activate a meaningful share of them almost immediately. What makes this system remarkable is its indifference to what's actually inside the stomach. A mechanoreceptor responding to stretch cannot tell the difference between a protein-dense meal and a glass of water because it isn't reading composition at all, only volume and pressure. That single fact explains a lot of what happens later in this video, including why some foods make you feel done long before you've eaten very much of anything. Once a gastric mechanoreceptor detects stretch, that information needs a way to reach the brain, and the pathway it travels is the vagus nerve, the longest cranial nerve in the human body, running from the brainstem down through the neck and chest into the abdomen. Electrophysiological recordings described by researcher Kentish and colleagues have shown that these vagal mechanoreceptors respond to stomach stretch rapidly and in a strikingly linear way, meaning the strength of the signal scales directly with how much the stomach wall is expanding. This isn't a vague or delayed transmission system. It functions closer to a dedicated cable running continuously between 2 points, carrying real-time information about physical distension for as long as food sits in the stomach. Research by Phillips and Powley in rats demonstrated that gastric distension suppresses feeding specifically through this vagal pathway, and that animals with these mechanoreceptors surgically disrupted go on to eat significantly larger meals, a finding later replicated in additional rodent studies using different methods to interrupt the same nerve pathway. The vagus nerve, in other words, isn't just background plumbing connecting the gut to the brain. It's the specific wire this entire early satiety signal depends on, and without it functioning properly, that first stretch-based message from the stomach simply never arrives where it needs to go, which raises an obvious next question. Where exactly does it arrive, and what happens the moment it gets there? The vagus nerve doesn't deliver its signal straight to the parts of the brain most people associate with conscious thought or decision-making. It terminates first in a small ancient structure in the brainstem called the nucleus of the solitary tract, or NTS, tucked deep in the dorsomedial medulla. Medical literature describes the NTS as the first relay station for visceral sensory information carried by the cranial nerves, meaning it's the very first stop this signal makes anywhere in the central nervous system. That detail matters more than it might seem to at first. It means the earliest processing of "I'm starting to feel full" happens in a region operating largely below conscious awareness, in the same general area responsible for regulating heart rate, breathing, and other automatic functions. A review by researchers Grill and Hayes described the NTS as a portal that integrates visceral afferent signals with the body's broader energy status, well before that information ever reaches the hypothalamus, or the higher brain regions involved in deliberate choice. In practical terms, the very first flicker of fullness you notice during a meal isn't a decision your brain makes. It's closer to a reflex being registered by a structure that evolved specifically to monitor what's happening inside your organs, moment to moment, without waiting for permission from anywhere else. And this mechanical stretch-based pathway turns out to be so sensitive that it can be triggered by something with 0 calories in it whatsoever. Since gastric mechanoreceptors only track stretch and pressure, not the chemical makeup of what's sitting inside the stomach, plain water can trip the exact same early alarm as a calorie-dense meal. A study published in Physiology and Behavior tested this directly by infusing liquid food into participants' stomachs through a tube, bypassing taste, smell, and chewing entirely, so that only the internal gastric mechanism was left doing the work. The design was elegant precisely because it isolated volume from calories. 2 infusions carried the exact same energy content, but one was diluted to a larger volume than the other. The larger, more diluted infusion produced a measurable drop in energy intake at the following meal. Roughly 77 fewer kilocalories eaten, about 13% less, even though both infusions had delivered identical calories beforehand. Volume alone, with energy held constant, was enough to change how much food came next. That result has a fairly direct real-world translation. Drinking water, or eating foods with high water content like soups, salads, or most fruit, can trigger the same mechanical fullness pathway, independent of how many calories those foods actually contain, simply because the stretch receptors respond to occupied space rather than stored energy. But this mechanical alarm is only the opening move. A second, entirely different kind of signal follows close behind it, and it changes the picture considerably. Before we go further, of everything we've covered so far, which part actually surprised you the most? Was it that blood sugar moves too slowly to explain early fullness? Or that a mechanoreceptor in your stomach can't tell water from a full meal? Drop it in the comments. I'm curious which one lands hardest for different people. While the mechanical stretch signal is still firing, a second messenger starts building in the background, cholecystokinin, or CCK. Its role in satiety was first identified in a landmark 1973 animal study by researchers Gibbs, Young, and Smith, who found that injecting CCK into rats caused them to stop eating earlier, in a pattern that closely mimicked natural satiety rather than sickness or discomfort. It's worth being clear that this foundational finding came from rodents, not people. Human confirmation followed less than a decade later. A study led by researcher Kiselev and colleagues infused CCK into human volunteers and found it reduced food intake by roughly 16%. Critically, the subjects didn't eat more slowly. They simply stopped earlier, which is exactly the pattern you'd expect from a genuine satiety signal rather than something that just makes eating less pleasant or more difficult. What makes CCK particularly interesting is where it acts. It's released from the small intestine in response to fat and protein breakdown products, and it binds to CCK receptors sitting on the very same vagal afferent fibers already carrying the mechanical stretch signal from the stomach. The two signals, one physical, one chemical, end up converging on a shared neural pathway rather than operating as separate competing systems, which means they reinforce each other rather than working independently. CCK isn't working alone for long. Specialized cells lining the intestine, known as L cells, release two additional hormones in response to the arrival of nutrients, GLP-1 and peptide YY. A study led by researcher Gutzwiller and colleagues, published in Gut in 1999, showed that GLP-1 potently reduces food intake in healthy human volunteers, and a companion study by the same research group, published the same year in the American Journal of Physiology, found a closely matching effect in patients with type 2 diabetes, meaning the response holds up even when insulin signaling itself is impaired. Peptide YY follows a similar script. Research by Batterham and colleagues, published in Nature in 2002 and later in the New England Journal of Medicine in 2003, found that infusing PYY into human volunteers reduced food intake by as much as 35%, with a comparable drop in total calories consumed. The effect held in both lean participants and participants living with obesity, suggesting the pathway itself stays intact even when body weight changes. Only the response to it may shift over time. Put the pieces together and a clear picture emerges. By somewhere between 10 and 20 minutes into a meal, the brain isn't relying on a single signal at all. It's receiving mechanical stretch information from the stomach wall, CCK from the small intestine, and now GLP-1 and PYY from further down the digestive tract. 4 separate lines of evidence converging on the same vagal to brainstem pathway, all pointing in the same direction. By the time someone would consciously describe themselves as getting full, that description is arriving late, after a genuine cascade of signals has already been building for several minutes. If the signaling system is this thorough, a fair question follows. Why does anyone ever eat past the point of fullness at all? Part of the answer showed up in a 2025 study published in Science, where researchers Zhu and colleagues identified a specific population of dopamine-releasing neurons in the brain's reward circuitry that activate specifically during the act of eating palatable food, not before it and not after, but precisely while consumption is happening. This research was conducted in mice. In the study, artificially boosting activity in these neurons during a meal extended how long the animals kept eating, mimicking exactly what happens when food is unusually palatable. The researchers connected this to something nutrition scientists have informally called the salted nut phenomenon. the observation that highly palatable food can sustain consumption regardless of an animal's actual physiological energy needs. Interestingly, activating a GLP-1 receptor agonist in the same study suppressed the activity of these reward neurons, suggesting the satiety and reward systems don't operate as a simple one-directional hierarchy. They actively compete. What this means in practice is that the arrival of a satiety signal at the brainstem doesn't automatically guarantee it gets acted on. A strong enough reward response, driven by how palatable a food is rather than how much of it has been eaten, can partially override or delay the behavioral response to signals that are, physiologically speaking, firing exactly as they should. The wiring for fullness can be working perfectly and still lose, at least temporarily, to a separate and very old reward circuit. Everything covered so far belongs to what researcher John Blundell's Satiety cascade framework calls satiation, the fast within-meal process that determines when a single eating episode ends. But there's an entirely separate signal running in parallel on a completely different clock, and it answers a different question altogether. Not should this meal end, but how much food does this body need over the coming days? That signal is leptin, a hormone secreted by fat tissue in direct proportion to how much of it a person is carrying. Its discovery was described in detail by researcher Jeffrey Friedman in a 2019 review published in Nature Metabolism, which characterized leptin as an afferent signal in a negative feedback loop, informing the hypothalamus about the body's long-term energy reserves, rather than the volume of any particular meal. The distinction matters because leptin doesn't rise and fall meal to meal. It shifts gradually over days, tracking changes in body fat rather than the last thing someone ate. That means the fast mechanical and hormonal signals covered earlier in this video and the slow leptin signal are, functionally, two different systems answering two different questions, even though both ultimately report to overlapping circuits in the brain. Bring all of this together and the sequence looks less like a single switch and more like a relay race with several distinct legs. Stretch receptors in the stomach wall fire first, within minutes, based purely on volume. CCK follows from the small intestine. GLP-1 and PYY arrive shortly after from further down the gut. All of it converges on the same vagal pathway into the nucleus of the solitary tract, arriving well before any of it becomes a conscious thought. But speed and volume of signaling only matter if there's time for them to register. Eating quickly can outpace how fast gastric stretch builds and how fast these hormones can rise, meaning a person can take in a meaningfully larger volume of food before the accumulating signal reaches a level the brain responds to. Layer a strong reward response from especially palatable food on top of that timing gap, and the physiological system, while working exactly as designed, ends up several steps behind the behavior it's supposed to be regulating. That's really the story this video set out to tell. Not a single dramatic signal, but a layered, largely unconscious sequence of mechanical, chemical, and reward-based information. Most of it happening in a part of the brain that has nothing to do with conscious thought at all. If this changed how you think about what actually happens between your first bite and that moment of feeling done, give the video a like. It helps this kind of research reach more people. Subscribe if you want more of these deep dives into the science behind everyday sensations, and send this one to anyone who's ever wondered why they felt full before their plate was even half empty.